Bismillahirrahmanirrahim. Assalamu alaikum. Dear students, today we are going to study remaining part of the bacterial genetics, and in today's lecture we will look at the mapping of bacterial genes with interrupted conjugation, and then a natural gene transfer and antibiotic resistance, and after this transformation in bacteria and bacterial genome sequences. So first of all we will look at the mapping bacterial genes with interrupted conjugation. So, the transfer of DNA that takes place during conjugation between high frequency recombinants and F minus cells, these are F minus cells, so allows bacterial genes to be mapped. So, when we interrupt the conjugation at different intervals, different time intervals, we can use this technique to map bacterial genes. So during conjugation, the chromosome of the high frequency recombinant cell is transferred to the F minus cell, and transfer of the entire E. coli chromosome requires about 100 minutes. So 100 minutes is the time needed to transfer the whole E. coli chromosome from HFR to the F minus cell. So if conjugation is interrupted before 100 minutes, before 100 minutes of time have passed, only part of the chromosome will pass into the F minus cells and have an opportunity to recombine with the recipient chromosome, because when part of the chromosome is transferred to the F minus cell, the recipient cell, then after the transfer, recombination takes place between the recipient chromosome and the transferred chromosome. So if there is no recombination, then cell will not take up the DNA, recipient cell, and the non-recombined chromosome will be degraded. So the chromosome transfer always begins within the integrated F factor and proceeds in a continuous direction. So genes are transferred according to their sequence on the chromosome, and the time required for individual genes to be transferred indicates their relative position on the chromosome. So in this way we can map the genes. Now we will look at a problem, and in this problem let us look at a cross analyzed by Francis Francois Jacob and Ali Wollman, who first developed this method of gene mapping. And they used donor high frequency recombinant cells that were sensitive to the antibiotic streptomycin, and gene for this streptomycin sensitivity is STRS. So resistant to these HFR cells were also resistant to sodium azide, AZI or gene, and infection by bacteriophage T1, and this gene is TONR. So prototrophic, these cells were prototrophic for threonine, leucine, and they were able to break down lactose and galactose. So these are the cells, and they use the F recipient cells that were resistant to streptomycin, and they were sensitive to sodium azide. So they used the F recipient cells that were resistant to the streptomycin, sensitive to the sodium azide, and to infection by bacteriophage T1, and auxotrophic for the threonine. Auxotrophic means that they cannot synthesize threonine and leucine, and they were unable to break down lactose and galactose. So, thus the genotypes of the donor and recipient cells were as follows. For the genotype of donor cell is STRS, THRF plus, leucine plus, sodium azide resistant, bacteriophage T1 resistant, lactose and galactose prototrophic. And the recipient cell genotype was, because it was a F minus cell, so the recipient cell was streptomycin resistant, uh, auxotrophic for threonine and leucine, and sensitive to sodium azide and bacteriophage T1 infection, and it was also auxotrophic for lactose and galactose. So, these are the two cells and HFR cells and F minus cells and their respective genotypes. So, uh, they used the interrupted conjugation for mapping the genes of these uh, two cells. So, in their experiment, they performed the conjugation, and they used the they interrupted the conjugation at different intervals. So, when they interrupted the conjugation and uh, grown the cell on different media and selected the cell on different media, then after 8 minutes uh, and 10 minutes and 16 and 25 minutes, different genes were transferred from one cell to the other cell. So, uh, they concluded that the transfer time indicate the order and relative distance between the genes, and can be used to, this time can be used to construct a genetic map also. Time, for example, in this figure, time is plotted on the x-axis, and percentage of these cells displaying particular trait is plotted on the y-axis, and the increase in time and increase in the percentage of cells having different trait is helpful in mapping the genes and their order on the bacterial chromosomes. So, for example, if we look at this time interval and the presence of trait in the bacterial cells when they were plated on the selective media. So, the AZI gene, sodium azide gene, was sensitivity or resistance gene was present on the was seen in after the 8 minutes, after 8 minutes of interval, approximately this at this time, and after this uh, around after 10 minutes TON gene, and then similarly LAC gene near 18 minutes, and GAL gene near 25 minutes. So, in this way we can interpret that which gene is coming first and which gene is coming later, and by growing the cells after different intervals of interrupting the conjugation, when they are grown into the selective media, they can be tested for different traits, and the traits which are appearing after um, after less time, they are uh, they are coming first, they are transferred first to the recipient cell, and the genes which are the traits which are present after longer time, they are transferred later. So in this way there is a directionality of gene transfer also. So directional transfer we can easily see in this figure because there are different cells which are used when we are plating different uh, cells after the interrupted conjugation. So different genes are present first, and different genes are present after more time. So if we look at the strain HFR1, so in HFR1 we can see that this F factor F factor is present between AZI gene and leucine gene, because chromosome is circular. So the transfer occurs uh, where the F factor is inserted into the recipient cell, and if we linearize this uh, chromosome, then we can see that leucine gene is transferred first in the HFR1 strain, and then threonine, thymine, histidine, gal, lac, pro, and AZI gene. But in the case of HFR5 strain, the F factor is inserted between the histidine and thymine genes. So in this case, uh, after the F factor, thymine gene is present, and similarly the other genes. So if we map, if we or look at the transfer of F factor and other genes in different strains, then we can find the order of the genes on the bacterial chromosome. So as we can see that in this figure, in the first figure, the order of the gene transfer is given, and it is unaligned, and it is based on the orientation of the F factor. So after the F factor in strain H, threonine gene is present. In strain 1, leucine gene is present after the F factor, and similarly in strain 2, proline is present. So it means that the F factor, when F factor is inserted, it is inserted between two genes, and it is a random uh, process. But uh, when we align these genes, we can also, when we look at the second figure, where order of gene transfer with aligned genes is given in different strains, orientation of insertion of the F factor is different. In strain 5 and strain H, it is F factor is followed by F factor is inserted between thymine and histidine, and in the strain H, F factor is inserted between threonine and thymine, and in the other four strains, 4, 1, 2, and 3, the orientation is different. And when we uh, look at the uh, orientation, then we can easily figure out that where F factor was inserted, then the first gene which appeared on uh, uh, after less time, after almost 8 minutes, it is the gene which is following the F factor. In the case of strain 5, it is thymine. In the case of H factor, it is threonine, and similarly in the strain 4, it is thymine. In the case of strain 1, it is leucine. In the case of strain 2, it is proline, and in the case of strain 3, it is lac gene. So in this way we can map the bacterial genes on the chromosome. So the order of genes on the chromosome is the same, but the position and orientation of the F factor differs among the different strains. So in this way the interrupted conjugation method can be used to map the bacterial genes. So next is the natural gene transfer and antibiotic resistance. So many pathogenic bacteria have developed resistance to antibiotics, particularly in environments where antibiotics are routinely used. And these environments are hospitals and fish farms. So massive amount of antibiotics are often used in the aquaculture to prevent the infection in the fish and enhance their growth. So when a massive amount of antibiotics are used, then it is causing the selection of the antibiotics, selection of the strains which are resistant to different antibiotics. So continual presence of antibiotics in these environments selects the bacteria that are resistant, and this reduces the effectiveness. And this thing, excessive use of antibiotics reduces the effectiveness of antibiotic treatment for medically important infections. So hospitals, and because hospitals are also the environments where mostly antibiotics are used, so they are the places where antibiotic resistance emerges. So antibiotic resistance in bacteria frequently results from the action of genes that are located on the R plasmid. So they are small circular plasmids that can be transferred by conjugation. So conjugation is the way of uh, spreading the resistance among different bacteria and bacterial species. So the results of recent studies have demonstrated that the R plasmids and their resistance genes are transferred among bacteria in a variety of natural environments. So in one study, uh, plasmids carrying genes for resistance to multiple antibiotics were transferred from a cow udder infected with E. coli to human strains of E. coli on a hand towel. A farmer used when he milked the cow with infected udder, and then he wiped his hands after uh, milking the infected cow, and then he might have unwittingly transferred antibiotic resistance bacteria from bovine to the human inhabiting microbes. So in this way the resistance genes are transferring from one species to the other species, and conjugation taking place in minced meat on a cutting board allowed plasmids to be passed from pig to the human E. coli. So, the transfer of R plasmids also occurs in sewage, soil, lake water, and marine sediments. So, these are all natural gene transfer in bacteria. So, perhaps most significantly, the transfer of R plasmid is not restricted to the bacteria of the same or even related species. And R plasmid with multiple antibiotic resistances have been transferred in marine waters from E. coli and other human inhabiting bacteria in the sewage to the fish, bacterium, um, Aromona salmonicida, and then back to E. coli through raw salmon chopped on a cutting board. So, all these are the natural ways of transferring resistance genes, antibiotic resistance genes from one species to the other bacterial species. So, these results indicate that the R plasmids can spread easily through the environment, passing among related and unrelated bacteria in a variety of common situations. So, that they can do so underscores both the importance of limiting antibiotic use to treat medically important infections. So, it is important and necessary to lower the use of antibiotic use for treating the medically important infections, because excessive use will result in causing the R plasmids to spread very fastly. And it is also highlighting the importance of hygiene and sanitation in everyday life. So, the cleanliness is a key to stop antibiotic resistance from spreading in the environment. So, it is very important. Otherwise, uh, the bacterial species are having different resistances developed, and our plasmids are containing the genes, and uh, excessive use of the antibiotics will results in selecting the resistant strains, because when antibiotics are used, the strains and species that are sensitive to those antibiotics will die, and only the strains that are having the resistance genes for those specific antibiotics will start growing at a very rapid pace, and in this way um, multiple resistances can be present in uh, different bacteria, and that can be very difficult to treat if this practice continues. As we already know that there are multiple resistances which have been developed in the Mycobacterium tuberculosis, and there are several other species. For example, Salmonella typhi is also a multi-drug resistant, uh, and in this way it hampers uh, mm, uh, all the sorry it stops the humans from developing good antibiotics that can be used to treat multi drug resistances. So it is better to uh, develop better practices, and uh, keeping the places and everything clean, and clean environment will result in hindering the development of antibiotic resistance. So now coming towards the transformation in bacteria, a second way that DNA can be transferred between bacteria is through the transformation. And transformation is actually take of DNA from the medium of the bacterial cell. So transformation played an important role in the initial identification of DNA as the genetic material, and transformation requires both the uptake of DNA from the surrounding medium and its incorporation into the bacterial chromosome or plasmid. So, it may occur naturally when dead bacteria break up and release DNA fragments into the environment, and in soil and marine environments. This means uh, may be an important route of genetic exchange for some bacteria. So, cells that take up the DNA and are said to be competent cells, and some species of bacteria um, that take up DNA more easily than some other species, and this competence is influenced by growth stage, the concentration of available DNA, and the composition of the medium. These are the factors on which the competence of the cell is dependent. So, the uptake of DNA fragments into a competent bacterial cell appears to be the random process, and DNA do not need even to be transferred uh, to the um, bacterial cell. Virtually any type of DNA, that can be bacterial DNA or any other DNA molecule, can be transferred to competent cells under appropriate conditions. As we can see in this figure that uh, double stranded DNA fragment comes across the bacterial cell, and only one strand of this double stranded DNA is taken up uh, by the cell, and when this is taken up by the cell, there are homologous sequences on this DNA fragment and the bacterial chromosome, and recombination takes place between this fragment and the bacterial chromosome, and the excised DNA fragment is then degraded. And when this cell further divides into two daughter cells, so one cell becomes recombinant because it is now containing the new DNA, but the other cell remains the non transformed or non recombinant cell. So, in this way, DNA is transferred to the bacterial cell, and this is the process of transformation. And just like interrupted conjugation, transformation can also be used to map the bacterial genes. So, for example, if this is a uh, donor cell and this is the recipient cell, so the DNA from a donor cell is fragmented. Fragments may contain one or more genes of interest. For example, A plus, B plus, and C plus genes are present in the donor cell, and when this bacterial cell is the DNA from the donor cell is fragmented, and uh, fragments are taken up by the recipient cell, then uptake of A plus, B plus, and C plus, and then uh, A and B can occur. And after entering into the cell, the donor DNA becomes incorporated into the bacterial chromosome through the crossing over. So, this cell is transformant, and it is containing the A plus gene, and the say this second cell is containing the B plus gene, and this third is containing the C plus gene. But in the case of this fourth strain, there are two genes, A plus and B plus. So, this strain is known as the cell that is co-transformant, and this co-transformant is containing A plus and B plus. So the genes that are close to one another on the chromosome, they are more likely to be present on the same DNA fragment, and the and be recombined together, and they can go together. So the conclusion is that the rate of co-transformation is inversely proportional to the distance between the genes. So if distance, it means that when uh, distance is more, the rate of co-transformation will be less, and when the distance between two genes is less, the rate of co-transformation will be more. So in this way they are inversely proportional, the rate of co-transformation and the distance between the genes. Now coming towards the bacterial genome sequences. So genetic maps serve as the foundation for more detailed information provided by DNA sequences such as gene content and the organization. And geneticists have now determined the complete nucleotide sequence of a number of bacterial genomes. And genome of E. coli is one of the most widely studied of all bacteria, and it is a single circular chromosome, single DNA molecule, approximately 1 millimeter in length, and it consists of 4.6 uh, million base pairs, and an estimated 4,300 genes are present on this circular chromosome, and more than half of these genes have no known functions. They have been they have been identified as genes, but their function is still unknown. So in most of the bacterial species, function of most of the genes is still unknown, and these orphan genes may play important roles in adapting to unusual environments, coordinating different metabolic pathways, and in the organization of the chromosomes, or they may be involved in communicating with other bacterial cells or species. So a number of other bacterial genomes have also been completely sequenced, and many additional microbial sequencing projects are also underway. So a substantial proportion of genes in all bacteria have no known function until now. So in this table, different bacterial species are given for which genomes have been sequenced, and their number of genes have also been predicted, and their GC content is also given, and in all these species uh, almost half of the genes identified are still having no identified function. So in this field there is still a room for research, and scientists are working on these species, because there are millions of bacterial species present on this earth, and only few thousands have been completely worked out, and because it is very difficult to culture and grow all the bacterial species, so only medicinally are important species, and those which are for the which are good for human and animals have been the subject of research. So I think around 8,000 bacterial species have been identified, and their uh, cultures, matter for culturing those bacteria have been identified, and still a lot of bacterial species still need identification. Uh, so this is the genome of bacterial E. coli bacterium. So E. coli bacteria has its uh, genome sequenced, and different uh, percentages of genes affecting various known and unknown functions are given. There are several different types of genes. So genes for fatty acid and phospholipid metabolism, for example, and transcription and RNA metabolism, and many other cellular processes have been identified. So this is the genome of E. coli. And similarly, the bacterium uh, Haemophilus influenza was the first free living organism which was sequenced, and this is the map of this bacterium, genome of this bacterium. So uh, it is also indicating several different genes which are involved in amino acid biosynthesis, biosynthesis of cofactors, and similarly cell envelope genes and many other different genes encoding different structural and functional enzymes and proteins and enzymes are, and still there are several unknown genes which are indicated by white color in this figure. So I think this is now complete for the bacterial genetics, and if you have any question you can ask on during the online question answer session. So thank you for today.